In urology clinics today, doctors routinely treat bladder cancer with a surprising method. They inject an old tuberculosis vaccine directly into the patient's bladder. This chart compares Alzheimer's rates. The left column shows standard treatment. The right shows the tuberculosis vaccine group, with a sharp 26% drop. Expanding to diabetes data, a downward line tracks p-tau217, the primary Alzheimer's biomarker, showing a steep reduction from multiple vaccine doses. Classical immunology states that a vaccine only trains the body to fight one highly specific pathogen. You get a measles vaccine to prevent measles, and a polio vaccine to prevent polio. An injection meant to prevent a lung infection should not shrink a bladder tumor, and it certainly shouldn't protect the brain from neurodegeneration. These cross-system effects suggest that the boundaries we've drawn around specific immunity are far more porous than our textbooks allow. In 1908, the French industrial city of Lille was struggling with a massive tuberculosis outbreak. A physician named Albert Calmette partnered with a veterinarian named Camille Guérin to study the disease. Guérin's background in animal husbandry meant he didn't look at illness the way a clinical doctor would. He focused on keeping entire herds healthy through prevention, rather than curing individuals who were already sick. They started by taking a deadly strain of bovine tuberculosis and growing it on a simple mixture of beef bile and sliced potatoes. As the bacteria adapted to this unusual food source, it lost its lethal characteristics with each passing generation. They decided to repeat this process, moving the bacteria into fresh cultures every 3 weeks, until it was weak enough to safely use as a human vaccine. They maintained this exact 3-week cycle for 13 years. They kept the cultures alive straight through World War I, even as German forces occupied Lille and turned the city to rubble. After 230 consecutive transfers, the fully weakened strain was ready. They called it Bacillus Calmette-Guérin, or BCG. 13 years of manual repetition in a war zone, driven by an agricultural approach to medicine, created what would become the most widely administered vaccine in human history. Following the Second World War, the Red Cross and UNICEF launched massive campaigns, administering BCG to tens of millions of children across Europe, Asia, and Africa. The United States and the Netherlands opted out. They needed to preserve the accuracy of their tuberculosis skin screening tests, which the vaccine would trigger. That decision created 2 distinct global populations, one vaccinated and one serving as a control group. Soon, strange data emerged. Vaccinated children survived childhood at higher rates, entirely independent of tuberculosis. In laboratory settings, mice injected with the vaccine saw their tumors shrink. The scientific community dismissed these reports as statistical noise. The prevailing dogma held that only the adaptive immune system, the T cells and antibodies, could remember specific threats. The innate immune system, the body's generalized first line of defense, was considered a blunt tool incapable of learning. Because researchers believed the innate immune system was untrainable, they spent decades overlooking evidence that BCG was granting a broad, non-specific immunity against multiple diseases. In 2011, an infectious disease physician named Dr. Mihai Netea demonstrated that innate immune cells do in fact develop a form of memory, a process he termed trained immunity. This epigenetic mechanism unwinds the architecture of an innate immune cell, leaving the specific genetic instructions for defense permanently accessible. The cell then produces generalized defense particles at a highly elevated baseline rate, ready to aggressively attack invading viruses, bacteria, or rogue cancer cells. This cellular reprogramming explains why the clinical data from the last 100 years looked so strange. It provides the biological pathway for how a lung vaccine could systematically impact both oncology and neurology. When this epigenetic reprogramming occurs, the immune system drastically increases production of an ancient antimicrobial peptide called LL37. Recent research at Stanford identified a crucial connection in the brain. Amyloid beta, the protein that clumps together to form Alzheimer's plaques, is an ancient biological sibling to LL37. LL37 acts as a natural chaperone. The 2 molecules wrap together, preventing amyloid beta from folding into toxic clumps. The entire sequence connects. BCG unwinds DNA, flooding the system with LL37, which locks on to brain amyloid, chaperoning it to keep the neural environment plaque-free. By boosting LL37, the vaccine appears to reinforce a natural pathway for handling amyloid beta before it becomes pathological. It's an ancient immune response, kept in a state of high readiness by a century-old bacterial signal. Two scientists, working through a military occupation with beef bile and potatoes, were simply trying to stop a lung infection in French infants. The fact that their century-old agricultural experiment may be our most actionable defense against Alzheimer's disease is an immense historical irony. It requires no new synthetic drug, just a very old organic reaction. It shows us the human immune system is deeply integrated, capable of solving biological problems long before we even have the vocabulary to name them.